In this session on visual physiology, we'll pick up from the previous sessions on visual anatomy and visual histology. In this session, we'll describe the structure and function of the photoreceptors, rods and cones, and identify how they are distributed in the retina. We'll describe the retinal circuitry. We'll describe the transduction by photoreceptors of light into neuronal activity. We'll describe the consequences of selective damage to rods or cones in various retinal diseases. We'll describe the inputs to the layers of the lateral geniculate nucleus in the thalamus, define the receptive field, and describe the receptive fields of cells within the visual pathway, such as the ganglion cells, the retinal ganglion cells, the lateral geniculate neurons, as well as the cortical neurons. And then we'll describe the anatomical and physiological properties of the magnocellular or rod system and the parvocellular or cone system. And finally, we'll identify the cortical regions representing color, form, location, and movement of visual stimuli in the world, and the deficits occurring when these regions are damaged, that is visual agnosia, prosopagnosia, as well as neglect. These keywords we'll be referring to throughout the session. We see here an artist's representation of the visual pathways. Whatever we perceive visually is dependent on these pathways. Visual input is detected by rod and cone photoreceptors distributed over the retina. These photoreceptors have very different sensitivity to the characteristics of light, and they have very different distributions over the retina. We see here that rod photoreceptors enable us to see even in dim light, while cone photoreceptors enable us to see in color in bright light. Rod and cone photoreceptors are named for the shape of the photoreceptor outer segment. This is a photoreceptor, a rod, and this is the outer segment. This is on a rod and a much smaller outer segment on a cone. The outer segment contains information that's going to transduce the activity of light, and it's made up of membranous discs that have photopigment. The inner segment contains the cell nucleus and most of the biosynthetic machinery that would be found in any neuron. And then there is a synapse that will connect with the next level of cell. We'd like to look at the differences between rods and cones, and then later on the neural systems connected to them. The rods have a very high sensitivity to light and are specialized for night vision. They have a tremendous amount of photopigment compared to the cones, and as a result, they can capture light more readily. They have high amplification so that they can actually detect a single photon, the least amount of light that can be presented. They saturate in daylight. That is, they become essentially non-functional in daylight. And they have a low temporal resolution, and that is, flashing lights would be fused if they were seen only by the rods. They would not be able to see rapidly flashing lights. And rods are very sensitive to scattered light that come even in from various directions. So it's not giving great information about location in the visual world. On the other hand, cones have a low sensitivity of light, and they're specialized for day vision. They have much less photopigment. They have much less amplification. They need more light in order to get an output. And they saturate only in intense light. They have a high temporal resolution, which means that they activate fast and they are stopped fast, and as a result, they can follow rapidly flickering lights. And they're much more sensitive to direct axial rays, and that helps to provide for good visual acuity. They're connected to a system. For the rods, we call it the magnocellular system, and for the cones, the parvocellular system, based on the size of the cell bodies of retinal ganglion cells that we'll be looking at shortly. This rod system has a low visual acuity because it has highly convergent inputs from many rods into a given neuron further down in the pathway. They are not present in the central fovea, and they're achromatic. This system provides no information about color. They have just one rod pigment. The cone system, on the other hand, has very high visual acuity, very little convergence of cones onto multiple different cells further in the nervous system. It tends to stay in a narrow track. And they are concentrated in the fovea. And they're chromatic. They have 3 different types of cones. Each cone has a distinct pigment that's more sensitive to one part of the visual spectrum than another. The retinal distribution of the photoreceptors is shown here. Cones are concentrated, as seen here, right at the fovea. We see the fovea represented at this location. It's the central portion of the visual field. And that's where a tremendous number of cones are located, and they drop off rapidly as you move away from the fovea. Rods, on the other hand, are absent at the fovea, but increase in number as you move away from the fovea. So that cones give a great deal of information about the central portion of the visual field. Rods give much more information about the more peripheral portions of the visual field. And then we see the blind spot where there are no photoreceptors. The blind spot is where axons from retinal ganglion cells leave the globe, and there are no photoreceptors at that site. The visual acuity over different parts of the retina in light and dark are striking in different levels of adaptation. If you're using your cones in bright light, the acuity is highest right at the fovea where they're concentrated. And it drops off rather rapidly as you move away from the fovea. As a result, we can read very well at the fovea and see things with good color. But as you move away from the fovea, you don't see things as well, and even with light adaptation, and you're not able to see high resolution or see color so well. On the other hand, in dim light, the rods are providing most of the information in dark adapted eye, and the cones provide less information, but at the midline, that is at the fovea, there is so little information provided by the receptors that you might not be able to see very well at the central portion of the visual field in the dark. The absorption spectra of the 3 types of cones reflect sensitivity to red, green, and blue wavelengths, and each cone has a preferential sensitivity to one or the other. All of the colors that we are able to perceive are built up by combining these 3 colors. Now we'll look at a number of retinal diseases. We start out with a friend who ignores you when you pass on the street. And when you stop him, he says he never saw you and must have been preoccupied by something. He says he can see fine, so you ask why he didn't come to the party last night. And he mentions that he's been avoiding driving at night. What could be happening here? The answer is that he has a defect of rod vision, such as retinitis pigmentosa. It impairs peripheral vision, and it impairs night vision. It may actually appear normal central portion of the visual field, but we would call that tunnel vision. Visual acuity in the central field may be perfectly normal until the disease progresses to involve the central portion of the retina. The disease is associated with a steady state turnover of photoreceptors in the outer segments that normally occurs, and those, because light is hitting these photoreceptors, they're recycled readily. And we shed those discs from the distal portion of the outer segment, and we manufacture new discs at the proximal region. Under normal circumstances, the discarded disc packets are readily phagocytized by the retinal pigment epithelium. In retinitis pigmentosa, a genetic disease, those packets of broken up discs are not properly phagocytized and they accumulate in the retinal space. Eventually, they'll produce retinal detachment and blindness. We have another individual, 75-year-old man, goes to the Department of Motor Vehicle Bureau to renew his driver's license and fails the vision test. He goes to the local optometrist who finds that his vision is not improved with different lenses and tells him to go to an ophthalmologist. The ophthalmologist finds that he has poor visual acuity and poor color vision. Looks in at the ocular fundus and tells him that he has macular degeneration. This fellow's never had an accident. Explain what could be going on and what would his visual fields look like? The answer is he has a defect of vision in the central part of his visual field, such as age-related or senile macular degeneration, sometimes called AMD. It's the most common cause of blindness in the elderly. It's due to a retinal dysfunction preferentially affecting cone vision. He may never have had an accident because cars are large objects and do not require the central field so much. Peripheral visual field loss is more of a problem for driving. The visual fields might show a relative bilateral central scotoma, that is a loss of vision in the central field. Peripheral vision may be normal. This can be treated. It's treated with an anti-VEGF. VEGF is the vascular endothelial growth factor. And intraocular injections with this anti-VEGF can be very helpful in protecting vision. And then finally, for retinal diseases, we have in microbiology, you and a friend are reviewing gram stain slides for a lab. He sees the organisms clearly, but can't identify them as gram positive or gram negative. Why could that be? The answer is that he has a color vision defect, and he's unable to differentiate red and green, the most common kind of color vision defect. It's a common genetic condition, affects about 8% of males. The gram stains are differentiated by color, not by shape. Visual acuity is usually unaffected. There's actually a normal number of cones and normal amount of photopigment, but the photopigments are not adequately differentiated, so the colors are not differentiated. We'd like to look at the transduction process. How does light become neural activity? It's dependent on two membrane protein molecules that define the unique physiology of the vertebrate photoreceptor. That is a visual pigment and a cyclic GMP gated channel. The visual pigment is a chromophore vitamin A aldehyde, also known as retinol. It's a G protein coupled receptor associated with controls the cyclic GMP phosphodiesterase activity. That absorption of light is determined by color vision, by this photopigment. The second protein is this cyclic GMP gated channel. It's kept open by cyclic GMP. It's permeable both to sodium and calcium ions, and in the dark, it's kept open by this cyclic GMP, and as a result, sodium is coming in all the time into this photoreceptor. So there is what we call a dark current, this very lively current through the sodium channel, even in the dark. And also calcium comes in through that channel. And if you're in the dark, if you move into the dark, you'll gradually adapt to that dark because calcium is also coming in through that channel. We'll look at the process that's involved in this phototransduction. And it involves closing these channels, this sodium channel, in the outer segment. And the steps are involved in the dark. The level of cyclic GMP in the outer segment, this cyclic GMP, is relatively high. So this cyclic GMP gated channel is open, allowing sodium to come in. That is the dark current. Phototransduction through this process produces closing of the channel and hyperpolarization. If you reduce the excitation, you get hyperpolarization. What's the process? Light strikes the photopigment, retinol, activating it and changing it from 11-cis to all trans retinol. The activated pigment stimulates a G protein, transducin, in the rods, which activates cyclic GMP phosphodiesterase. The cyclic GMP phosphodiesterase catalyzes the breakdown of cyclic GMP to 5' GMP. As GMP is loaded, as the cyclic GMP is decreased, the channel closes, thereby the photoreceptor becomes hyperpolarized. In the dark, due to the depolarizing current, the photoreceptor is constantly releasing a transmitter, glutamate, at their terminals. Light-induced hyperpolarization reduces the release of glutamate. Notice that the photoreceptors are the only sensory receptors that hyperpolarize in response to the relevant stimulus. They're a unique photoreceptor in that regard. This process of phototransduction is described in words here that are written out for you to review as you would like. We'd like to look at the retinal circuitry. Light travels in from the pupil through the vitreous and hits the retina. And when it hits the retina, it activates photoreceptors, and we see the photoreceptors here. And we see that there are both cones and rods, photoreceptors, rods and cones. And then photoreceptors are connected to bipolar cells that we see right here. And the bipolar cells connect the rods and cones to ganglion cells. So the direct pathway through the retina is light hits the photoreceptors, activates photoreceptors, which activate bipolar cells, which activate ganglion cells, and that goes into the thalamus. There are other horizontal cells and amacrine cells that provide lateral interaction to provide enhancement of local contrast so that we can see edges very well. If we look at a low-res view of the retina, we would have the vitreous right here, and we're going to look at this segment of the retina. And blown up, we can see the various layers of the retina next to this schematic of the retina. So light comes in through the vitreous. It activates these photoreceptors, the outer segments of the photoreceptors, which we can see over here. The cell bodies of the photoreceptors, cones and rods, we see here that are represented in this region. And then we see the outer plexiform layer where there are processes of the photoreceptors as well as of other neurons. And then we see the inner nuclear layer right here that includes the cell bodies of bipolar cells as well as horizontal cells and amacrine cells. And then we see the inner plexiform layer, that has all of the areas where many of these processes are located. And then the ganglion cell layer, where the large cell bodies of ganglion cells are sitting. And they will send their axon out to form the optic nerve, travel eventually up to the lateral geniculate nucleus. Those cells at various levels of the visual pathway have receptive fields. That is, it's a portion of the receptor surface, in this case the retina, which when stimulated activates the neuron. In the visual system, every cell has a receptive field, as they do in every other sensory system. And in this case, it's a receptive field on some part of the retina. And in that part of the retina, the cell will be, in this case, for a ganglion cell, it might be excited when the central portion, if this is a portion of the visual world, it might be stimulated, that is excited, when a central portion of this portion of the visual world is lit up, and it might be inhibited when the surround, that's a very common receptive field early on in the visual pathway, center surround, where the center might be excited, the surround might be inhibited, or the other way around, the center might be inhibited, the surround might be excited. These are the beginning building blocks of visual perception. And there may also be color antagonism, where the center is excited of a particular cone. The center is excited by one color, and the sound, the surround stimulation would inhibit the cell. And they might, depending on if there was only the center was activated, the cell would be excited. If the surround was activated, it would be inhibited. So it would be most sensitive to a very small stimulus in the center. Whereas if there was a big stimulus, the negativity might counteract the positivity and there would be no response of this cell. So it's going to be very sensitive to things that have a very small amount of light in a small region. As we move into the central visual pathways, we can see that the two eyes are aligned in such a way so that input from one point in the visual world arrives at both retina, and that input will travel back through the optic nerves, the chiasm, and then from the two eyes, they will travel together in the optic tract and arrive at the lateral geniculate. The cells in the lateral geniculate will sort themselves out so that those that are coming from, mostly from cones, will arrive in certain layers we call the parvocellular pathway or the P channel. And those that are coming from rods, we call the magnocellular pathway or the M channel. Those cells, those inputs will synapse in the lateral geniculate, and then cells in the lateral geniculate will send their fibers on to the primary visual cortex, area 17 or the striate cortex. The cortical neurons have a very different interest. They are primarily concerned with edges, such as this, and contours. And as a result of what transformation has happened from the retina all the way back to the lateral geniculate and the cortex, we now have cells in the cortex that have input from both eyes, and they're preferentially interested in lines and edges. And as a result, when we look at an object like this, we build this world primarily out of lines and edges. So we might see this as a triangle, even though there is in fact no completed triangle here. And the same with this, we see as a triangle. It will have an effect on our perception. The sensitivity of cortical neurons defines what we will see. We see another, for instance, here in the work of an artist or a cartoonist. They long appreciated that simple lines provide powerful information about objects. And in fact, camouflage utilizes this property of the brain, of visual cortex, to hide objects. So if we look at the next object, we'll see that it's something that's very difficult to detect. And that is, if we look at this, it may be impossible to discern until you have pointed out that there might be a dog right here that's walking along a pathway with its head right here and its legs right here. And there might be a tree over here that has a shadow here. Camouflage is effective because it has taken away all the straight lines. And our visual system is searching for straight lines. So it just barely can make out this tree shape. And can barely make out the circle around this region. We are dependent on lines primarily in our visual world. If we look beyond the primary visual cortex to the pathways that travel from the primary visual cortex to other regions, we see a continuation of the information from the magnocellular pathway going up to the parietal occipital area. And this location carries information about motion, movement of objects, and the location of objects in the contralateral visual field from this hemisphere. So if this is the left hemisphere, it would affect the ability to see movement or the location of objects in the contralateral in the right hemifield. Damage to this area, in the parietal occipital cortex, results in neglect or deficits in spatial orientation. The neglect syndrome involves patients being unaware of what's going on. They don't recognize what's going on in the contralateral visual field. They seem unconcerned about what's going on in that field, even though they do not have a visual field deficit. Similarly, the pathway coming from the lateral geniculate to the primary cortex and subsequently to the visual association cortex in the temporal occipital region, particularly the inferior portion of the temporal occipital area. That tissue is involved in detection of color of objects and of the shape or form of objects. Damage to this region results in a visual agnosia, inability to recognize objects or colors. And it may be specific for faces, in which case it's called prosubagnosia, or to recognize inanimate objects, or to recognize colors, despite having no visual field deficit in the contralateral side. Where do you go from here? Individual neurons within the inferior temporal lobe of monkeys have been identified that fire maximally to faces, particularly faces of conspecifics, that is to other monkeys, whereas they give very little activation to random lines or to a hand. That is, they seem to be depending on input converging from the primary cortex into this temporal or temporal occipital region in order to see very much higher organized images. Where do we go from here, from the secondary association cortex for vision? In order to have that information be responded to emotionally, that information needs to travel from the association cortex to the limbic system in order to generate an emotional reaction.